next we have Dr. Kevin Magnum from the University of Michigan presenting IFN-beta regulates abdominal aortic aneurysm formation by modulating expression of the histone methyltransferase, SETDB2.
All right, thank you for the opportunity to present here today. Nothing to disclose. So abdominal aortic aneurysms are characterized by a number of morphological features, including loss of vascular smooth muscle cells, as well as increased expression and activity of the matrix metalloproteinases. And these within the abdominal aortic aneurysm wall are inhibited by tissue inhibitors of MMPs or TIMPs. Our lab, in the lab of Dr. Kathy Gallagher at the University of Michigan, studies a variety of chromatin-modifying enzymes that regulate the processes of various vascular surgical diseases, one of which is AAA formation. And one of the CMEs, or chromatin-modifying enzymes, that our lab studies is SETDB2. SETDB2 is a histone methyltransferase, and it silences gene expression by depositing the repressive trimethylation mark on histone 3 at lysine 9. And this ultimately leads to repression of gene expression of a number of inflammatory and downstream genes. So previous work from our lab has shown that SETDB2 was significantly upregulated in whole aortic tissue in a mouse murine model with elastase in abdominal aortic aneurysms, as well as in bone marrow-derived macrophages. And then recent data from our lab has also showed that in our single-cell RNA-seq database that specifically in the monocyte macrophage population, SETDB2 is significantly upregulated compared to controls. Recent data from our lab has also showed, as background, that when you deplete SETDB2, mice have smaller aneurysms in a chronic angiotensin model. And we showed that this was through significant upregulation and expression of the tissue inhibitors of MMPs, or TIMPs. And this is a family of conserved proteins 1 through 4, and all of these were significantly upregulated when you deplete SETDB2, specifically in the macrophage cell type. And the way that SETDB2 was mediating this was that it was, in the absence of SETDB2, there was decreased depositing of this repressive histone mark, H3K9 trimethyl, which we showed in chip assays at the TIMP1 promoters. So just to review, with increased expression of the TIMPs, you have decreased MMP activity, which was present when you deplete SETDB2 compared to wild-type controls. And this is just the schematic. Increased SETDB2 leading to decreased TIMP expression, increased MMP activity, and ultimately increased AAA progression. And so my question was, what are the upstream and potential downstream transcriptional mechanisms that are regulating expression of SETDB2? And taking a step back, we know through much research that chromatin states exist in a closed or open conformation. When the chromatin is closed, the gene transcription is turned off. And when the chromatin is in an open configuration, this allows transcription factors to access the DNA binding regions and increase gene transcription. And this is a very simplified view, but helps to kind of frame the model for the talk. And so my approach was to essentially look at open chromatin sites within the SETDB2 promoter to identify potential regulatory elements that regulated SETDB2 expression. And so what we have here at the bottom of the screen is the gene architecture of SETDB2 for the human SETDB2 gene. And the encircled aspect on this plot are open chromatin regions that align with DHS, or DNase hypersensitive regions, which are open chromatin regions in the genome. And not surprisingly, these also align with areas of active histone marks, such as H3K27 acetylation, as well as H3K4 trimethylation. And notably, there were a variety of conserved sequences within this promoter region. So compared to our bioinformatic analysis using this track from the UCSC Genome Browser, which is a publicly accessible database, we coupled this with in silico analysis of the SETDB2 promoter and found conserved STAT3 binding sites in the SETDB2 promoter. And specifically, there were 24 predicted STAT3 binding sites. And we were really excited by this because, as my colleague Dr. Adu mentioned, the STAT3 transcription factor is heavily implicated in regulating downstream inflammatory gene expression. However, to validate this in silico analysis, we used chromatin immunoprecipitation at the SETDB2 promoter to show that there was enrichment for STAT3 compared to IgG negative control. And a brief model of the interferon beta STAT pathway, we know that interferon beta binds to the interferon alpha beta or IFNR receptor. This activates the JAK tyrosine kinase receptor to phosphorylate STAT proteins, which then translocate into the nucleus, where they bind consensus elements to regulate downstream gene expression. And our lab has also used the chemical inhibitor tofacitinib to inhibit the JAK kinase and inhibit this pathway. So to interrogate the interferon beta pathway further, we used several approaches. We used genetic deletions or loss of functions mouse models, as well as pharmacologic inhibitors. So the plot on the left here shows that when you deplete STAT3, specifically in macrophages in a mouse model, they have decreased SETDB2 expression in macrophages. And then when you inhibit the interferon beta pathway with tofacitinib, you also have significant reduction in SETDB2 expression. Alternatively, when you stimulate bone marrow-derived macrophages from wild-type mice with interferon, you get a significant upregulation in SETDB2 expression. And then when you knock out the interferon beta receptor altogether in mice, you have a significant reduction in SETDB2 expression. So in conclusion, our lab had previously shown that SETDB2 was significantly increased in whole aortic tissue as well as macrophages from aneurysmal tissue, and that when you delete SETDB2 in mouse macrophages, this decreases aneurysm formation via increasing the protective TEMP protein expression, ultimately leading to suppressed MMP activity, which are those proteins that degrade the aneurysmal tissue. And then the novelty of the current study is showing that interferon beta, which is present in the aneurysmal macrophage cells, increases STAT3 expression as well as activity, which binds to the SETDB2 promoter to increase its expression. Increased SETDB2 expression represses the TEMP promoter activity, leading to de-repressed activity of the MMPs, thereby further promoting abdominal aortic aneurysm formation. I'd like to acknowledge all the members of my lab, including the support of my funding sources and that of Dr. Gallagher, and I'll happily take any questions.
It is very fascinating to see the overlap between some of the diabetic models and the aneurysm models between your two labs and the IL-17 and STAT3. So any questions from the audience? If not, thank you.
All right. Thank you.